Hello, everyone. Last time we talked about accessory organs. Okay, so this was the last slide we talked about last time. We talked about the CCK, which is a hormone from small intestine, which can increase the bile release. And bile is used for fat digestion. So today we're going to focus on the digestion and absorption. Okay, so in terms of the physical digestion, we're talking about, let me get to the slideshow here. In terms of physical digestion, we're talking about you break down big particles into smaller ones, but the chemical properties do not change, right? But if it's a chemical digestion, you actually change the chemical properties, right? So the list here are the types of chemical digestions. So for example, if you use a protein, if you digest the protein, you can make smaller peptides, and then smaller peptides, if you digest it further, you can make a single amino acids. Okay, so for lipid digestion, I'm going to talk about this later. But for carbohydrate chemical digestion, if you remember, in 430, you probably learned this. Polysaccharides can be digested into disaccharides and then can be digested into monosaccharides. All right. So if we start from the mouth, we talk about the digestion and absorptions in the GI tract. We start from mouth and then esophagus, stomach, small intestine, large intestine in that way. So we'll talk about these processes in each location in the GI tract. Okay, so in the mouth, all right, the pH is from 6 to 8. The physical digestion, can that happen in the mouth? Absolutely. So you have teeth to tear apart the big particles. You have the tongue to help you, mix the particles in with the saliva. Okay, and also help to break down the big particles. So those are physical digestion. How about the chemical digestion in the mouth? Remember, we know there is a enzyme in the saliva, salivary amylase, which can help to digest, which can chemically digest carbohydrates. Okay, so for chemical digestions, only carbohydrates can be digested in the mouth because of this amylase. Okay, in terms of absorption, there's nothing can be absorbed in the mouth. When we talk about absorption, we actually focus on the nutrient absorption. The nutrients are single sugar, glucose, and fatty acids and the small fat molecules. Those are considered as nutrients. So there's no nutrients can be absorbed in mouth. Okay, but if you think about other stuff, like say if you have a heart attack, if they put some nitroglycerin under your tongue and then can get absorbed into blood right away to help release the symptoms, right? So you do, if there's a lipid soluble vehicles carry something, and then it can be absorbed into blood, all right? But in normally, okay, nothing can be will be absorbed in mouth. How about esophagus? So if we talk about esophagus, it's pretty long. If we divide the esophagus into upper, middle, and the lower parts, remember the lower part, okay, the last 1 inch will penetrate the diaphragm and connect it to the stomach in the abdominal cavity. Okay, so this esophagus, mostly it is a path for food particles. Okay, no digestion, no absorption take place here. And if you remember, we talked about esophagus, very minimum amount of glands, the mucus glands are associated with this part of a tubular structure. And if there are any, they are mostly located on the distal end. Okay, in terms of helping to move the particles down, esophagus tubular structure, we do have muscular tissue around it. On the upper part, mostly skeletal muscles. Okay, skeletal muscles still like kind of extending from the larynx part. Okay, so esophagus, the skeletal muscles are mostly associated with the upper part of esophagus. In the middle part, in terms of muscles, we have both skeletal and smooth muscle cells around. The lower end, mostly smooth muscle cells. Okay, so here in the esophagus, no digestion, no absorption can take place. Now we'll get to the stomach. Okay, remember, stomach wall has 3 layers of smooth muscles in the muscularis externa. So the strong muscle movement can help to tear down the food particles. And also we have rugae, the rough surface inside of the stomach, which can enhance the physical digestion process as well. So stomach is a major organ for physical digestion. How about the chemical digestion? Is there any chemical digestion take place in the stomach? If you remember this enzyme, pepsin, pepsin is a protease, okay, which can digest the proteins. All right, so remember this pepsinogen can be converted to pepsin when hydrochloric acid is present, right? So we can say in the stomach, proteins can be digested. How about carbohydrates? No, there's no enzymes for carbohydrates in the stomach. Okay, and we say most of our enzymes, they function in pH close to your blood pH, 7.35 to 7.45. But in the stomach, the gastric juice is very acidic. If you look at the pH, is about 2 to 4, very acidic. So in this acidic environment, only pepsin can function, because pepsin is a very specific enzyme which can be activated in the acidic environment. Okay, so anything you eat in the mouth, you swallow. Okay, and if like say for example, a piece of bread you chew in the mouth, and then the amylase, salivary amylase, can partially digest this polysaccharide, and then you swallow and get into the stomach, and these polysaccharides, partially digested, they cannot be further chemically digested in the stomach, so they have to wait until they get to the small intestine, and then to continue the chemical digestion for polysaccharides. But for proteins, okay, in the mouth, there's no chemical digestion. But once in the stomach, then it can start the protein digestion, chemical digestion. Okay. gastric juice is very acidic. This is in our favor because, when you eat stuff, like especially if you eat a salad, for example, there are a lot of bacteria, in this uncooked food. So once you swallow the food particles into the stomach, because the gastric juice is very acidic, this will help to destroy all these pathogens. Okay, yeah, if you learned microbiology already, most of the common bacteria, they cannot tolerate acidic environment very well. So this gastric juice is a protective mechanism for us. Okay, in terms of absorption in the stomach, there's no nutrient that can be absorbed in stomach, but other things like a lipid soluble stuff, like just like the nitroglycerin we talked about in, if you put under the tongue, if it's a lipid soluble, then it can be absorbed. And also on top of that, alcohol can be absorbed in the stomach. So this is interesting. Let's say, if you go to a party, you start drinking some beer, and then, if you don't eat anything, you just have an empty stomach and you drink alcohol, and then you will pass out pretty soon, all right, pretty fast, maybe one beer, and then you're out. Talking about having a good time and you didn't, right? You passed out. So what should you do? You should, because alcohol can be absorbed in the stomach, if you have an empty stomach, this absorption process take place really fast, right? So before you go to the party, if you just drink some milk or eat some food, then this food particles can form a coat inside of your stomach, kind of on top of the lining cells. Then you start drinking some alcohol, and the alcohol will take a much, much longer to actually penetrate this coat, food coat layer, and get absorbed by stomach cells, right? So the point is, I'm just trying to help you to remember alcohol can be absorbed in stomach. Okay. In terms of regulation of digestion and absorption in the stomach, hormone can be regulate this process, and autonomic nervous system regulate the digestion and absorption in stomach as well. So this is the diagram you have in your book. Let me see where I can find it. It's on page 890. Okay, so it is the mechanism that regulated release of gastric juice. So for it to enhance the digestion in the stomach. So this part, I'm asking you to read your book and understand the process. Okay, so I'm going to focus on the chemical digestions in the stomach, in the small intestine, more. Okay, so now we're actually at the small intestine, the digestion and the absorption process in the small intestine. Okay, so we're going to focus on protein digestion and fat digestion and absorption. Okay. And for this part of the materials, I'm going to use the whiteboard. Okay, let me do this. Okay, so let me have the whiteboard down here. All right, so we're talking about the digestion take place in small intestine. So first is the protein digestion, okay? Protein digestion. Okay, so we're talking about the chemical digestion for proteins, right? So proteins can be digested into peptide and then being digested to amino acids. Okay, so the enzymes which can digest the proteins are called proteases. Okay, so if you remember, there are two proteases I talked about the last time. It's made in the pancreas. If you remember them, one is called trypsinogen. All right, the active form is called trypsin. Okay, another one is called. Did I give you this? I think I did. Procarboxy peptidase. Okay, this is the inactive form. An active form will be the carboxy peptidase. Okay, so now if you remember, the trypsinogen is made in the pancreas and it has to travel to the small intestine to be activated. The enzyme which activate the trypsinogen is called, right here, enterokinase. All right. So this enterokinase is a enzyme which can activate trypsin. This enterokinase is a small intestine enzyme. Okay, it's a protease made in the small intestine. So once the trypsin is activated, this trypsin is used to activate this procarboxypeptidase. All right, so you have the active carboxypeptidase to digest the protein in the small intestine. Okay, there's another enzyme from pancreas. It's a protease. It's called chymotrypsinogen. Okay, now it's nogen right here is inactive. Okay, and then this one, it can be activated to make the chymotrypsin. All right, the enzyme which can activate this enzyme is trypsin as well. Okay, so three proteases from pancreas. Okay, all right. Remember last time I asked you to start making a list of enzymes. The first one is the salivary amylase, and then the second one should be the pepsin, which is made in the stomach. And then now you have these three, trypsinogen, procarboxypeptidase, and chymotrypsinogen. These three are from the pancreas. Okay, now this one, maybe let me change the color. This one, enterokinase, is an enzyme made in the small intestine. All right, So on your list now, you should have 1, 2, 3, 4, 5, 6. 6 already. Okay, the first one is salivary amylase. The second one is the pepsinogen or pepsin. Either way will be fine. It's the same enzyme, right? And the third one should be the trypsinogen or trypsin. The four, it will be procarboxypeptidase or carboxypeptidase. Either one will be fine, okay? Number 5 is the chymotrypsinogen or chymotrypsin. And number 6 is the enterokinase, right? Okay, so we have 6 enzymes so far. All right, so these are proteases, which can digest the proteins, right? And so the proteases are from the pancreas and stomach and the small intestine. Okay, so now in the small intestine, we're still talking about the proteases. Okay, in the small intestine, one enzyme we talk about is the enterokinase. What is the function? To activate trypsinogen, right? Okay, and also in the small intestine, we have other proteases. Okay, one is called, let me give you the list. I want to follow your book. I don't want to give you more than your book. Let me see. All right, so maybe we do this. Okay, I'll give you the major ones. Okay, one is called aminopeptidase. That's a protease. Okay, another one is called a carboxypeptidase. Okay, this one is the same as from the pancreas. And then we have something called a dipeptidase. All right, these are the major proteases from small intestine. All right, so let me kind of explain this for a second. So if you think about a peptide is linked by amino acids. Okay, so amino acid number 1, amino acid number 2, amino acid number 3, and keep going. Okay, so then once you have the amino acids linked together, it's called a peptide, right? And then the peptide, have a specific three-dimensional folding to make a functional protein. So on this peptide, usually you have two ends. Not, you always have two ends. One end is called amino end. Okay, and this end will be the carboxy end. Okay, so if the enzyme is breaking down the bonds from the amino end, like from here, okay, so then it's aminopeptidase. Okay, if the enzyme is breaking down the bond from the carboxyl end, it should be carboxyl end, then it's called a carboxypeptidase. Okay, but if you only have two amino acids linked together, then you have an enzyme to break it down. So it doesn't matter if it's from amino end or from the carboxy end, because you only have two amino acids here. In this case, the enzyme here is called a dipeptidase. You got it? Okay, so in the small intestine, there are 4 important proteases I gave you. Okay, so you should have them on your list. Yeah, and at the end, I will give you the whole list. Okay, All right, so that's the proteases. Now let's look at the enzymes for still the small intestine. Okay, small intestine. We're talking about enzymes for carbs, carbohydrates. Okay, so here I want you to know three important one. One is the sucrose, 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 which is your table sugar, okay, which can break down to form glucose, simple sugar, plus a fructose. I hope you learned this in chemistry. These two are simple sugars, okay? When they combine together, they make a sucrose molecule, which is your table sugar, okay? So break down sucrose into these two simple sugars. This enzyme is called sucrase. This is the enzyme from small intestine, okay? Second one is the disaccharide, is called maltose. All right, this is the sugar you can find in the grains, like in the rice, in the flours, okay? This one can break down into two glucose molecules. All right, this enzyme is called maltase. All right, that is the enzyme from the small intestine as well. The third one is the disaccharide lactose, which is the milk sugar, which can break down to glucose plus the galactose. All right, so that's a simple sugar as well. So the enzyme is called lactase. So you all know this one, right? Lactase is the enzyme to break down milk sugar. So these are the three important enzymes I want you to know from small intestine. Okay, now we can now we can talk about the fat digestion. This is the part I need to spend a little bit more time. Okay, so when we talk about fat, we're talking about triglyceride. Okay, so triglyceride is made by um, the 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 glycerol right here, glycerol, let me write it down. We call it a backbone. It's a three carbon linked together, and then linked, you don't need to know the specific structure, but linked with like zigzag tails, okay? So these tails is called fatty acid. All right, so this zigzag kind of thing like that. At each corner is a carbon, 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 carbon. Okay, so there are a lot of these chemical bonds here, in the triglyceride. So these two together right here is called a triglyceride. That's what we call fat. Tri means the three fatty acid tails linked to the glycerol is called a triglyceride. Okay, so because there's so many chemical bonds in the fatty acids, that's why we say fat can store a lot of energy. So when you break down these chemical bonds right here, you can release energy for making ATP. Okay, so the triglyceride, when you eat a steak, a lot of fat in there, but you swallow this triglyceride into the stomach, there's no chemical digestion there because there's no enzyme for the fat in stomach. Okay, but you can physically tear these big fat particles to the smaller one. Okay, and then this triglyceride particles will get into the small intestine. So only in the small intestine the triglyceride can be chemically digested. So let's say these triglyceride molecules coming in this big particle, okay, and the enzyme which can chemically digest the triglyceride is called a lipase. Okay, so lipase can actually break down these triglyceride molecules like that. Okay, let me try this. Okay, so let me use a different color here. Okay, so this lipase can actually break down these bonds. If you only break down one, like say here, then you make the diglyceride. If you break down two, then you make monoglyceride. So the individual tails break down. These are the fatty acids. We already know, right? Okay, and then the leftover structure right here is called a monoglyceride. So if the triglyceride is chemically digested into the monoglyceride and the fatty acid, and if further break down this one as well, so you only have a glycerol in those forms, then these materials can be absorbed. But if it's in the triglyceride form, it cannot be absorbed. It's just too big. Okay, so for fat digestion, the first part is to make this triglyceride in to the form of fatty acid, glycerol, and monoglyceride forms. Okay, so I did say the enzyme which chemically digests the triglyceride is called a lipase. So let's say if you have a big fat particle, this particle is made by many triglyceride molecules, and then you have the enzyme get on the surface, and then the enzyme can only work on the surface. The center part is hard for the enzyme to get on for the chemical digestion. For that reason, we want to break down these big fat particles into smaller ones. Okay, so somehow if you can break it down into the smaller particles, and then the enzyme can actually be more efficient in terms of chemical digestion. Does that make sense? Okay, so just think about a process. Let's say if you have a bowl of water and you put 10 drops of cooking oil in the bowl and then let it sit for a while, all these droplets are going to merge together to become one big drop, right? That's kind of like this, all right? And now if you have a chopstick, all right, you stir really fast, then you can break it down into these many droplets. All right, so that's a physical process. You're breaking down these big particles into smaller ones, but the chemical properties do not change, right? So we call this process as physical digestion. So fat is first physically digested from big drop into smaller droplets, and then chemical digestion can be more efficient. Okay, so this process, the physical digestion of fat, requires bile to help. Okay, so let's talk about the bile here. All right, so now you have a big drop of fat. So when we say fat, it's triglyceride, right? Okay, So now you have a bile. Bile is a molecule which has the two parts. Okay, the head has no charge, but the tail has negative charge. Okay, so if the head has no charge and the fat triglyceride has no charge, non-charged molecules, particles, they have attraction force. They attracted to each other. So all this head, non-charged head, kind of attached on the surface of this big fat drop, and the tails kind of facing outside, kind of like that. Okay, so now in between here, of course, we have water, so it's a watery fluid around. Okay, and so if you have a lot of this same charged tail next to each other, close by, same charges repel from each other. So there is a force actually push them apart. And also with the help of water, now this fat drop can be teared into smaller droplets. Okay, this process is called emulsification. All right, you have that word in your book, emulsification. Emulsification is a physical digestion process. This is why the bile is needed for fat digestion. It's not for chemical digestion, it's for physical digestion. Okay, so maybe you have this, I'm sure you have this experience, in your kitchen, if you have a very greasy plate, if you use hot water, you can clean the dish much easier than using the cold water. Why is that? Because hot water is going to help you to break down these big fat particles into smaller one. You actually put extra energy, the heat, to break down the fat drop into smaller ones, and then you can rinse it off. Okay, same principle for detergent. Okay, so what is the function of detergent? It's very similar to bile. Okay, so to tear these big fat particles into smaller ones, and then you can rinse it off, right? So in our digestive system, we use bile to do the same thing. Okay, just tear this big drop of fat into smaller droplets, and then the lipase can work on these little droplets, chemically digest the fat. Okay, so lipase is from pancreas, if you remember that. All right, so let's talk about this chemical digestion then. All right, so if we have a triglyceride, now you have lipase. Now it's a chemical digestion. Okay, so you can break it down into different products. One is have the mono. One has one fatty acid tail. It's called a monoglyceride, and then you can have some just the glycerol, and then you can have some free fatty acid. Okay, so you can break it down into different products. So if you have the fatty acid right here, fatty acid, and also there's some phospholipid in your system you're going to use, is called a lecithin. Okay, so these are the phospholipid. Okay, we have in our small intestine, from food digestion. And also there are some cholesterol, and On top of that, okay, so on top of this, and then we have bile. Okay, so this bile right here is bile. Okay, so then you can form this kind of little structure. This structure is called a micelle. If you heard about this one before, um, let me kind of explain this. Okay, let's say if you have phospholipid, okay, when you learn about the uh, cell membrane structure, remember, is made by phospholipids. Okay, so these phospholipids, they have the head and tail. The phospholipids, the head, there's no charge, so they kind of go together. All right, they have a force to attract each other. And the tail, they have the negative charges, they're facing the watery fluid. This is a relatively stable structure. Okay, that is called a micelle. Okay, micelle. All right, so it's relatively a stable structure comparing with something like this. Okay, lining up just individually. All right, so kind of like if you have drops of cooking oil in water, then these drops, they tend to go together to form these little micelles. All right, so they have these non-charged heads facing each other and the charged tails facing outside. Okay, that's a micelle structure. So in terms of the fat chemical digestion, you break down triglyceride into fatty acid. I did say that, right? Fatty acid and maybe the some of the mono uh, glyceride, okay, and also there's some cholesterol and some of the phospholipids, lecithin, and then you have all this being included into uh, uh, in the center and surrounded by bile. Remember, bile has the head and tails. Head has no charge and tail has charges. So this whole structure now is forming a micelle. Okay, so once you have this micelle, all right, it can be moved into the small intestine cells. Let me draw the small intestine lining cells here. Okay, this is the lumen. Okay, the food, the chyme is moving that way. Okay, so this is the simple columnar cells with microvilli. Okay, so once you have this micelle, then this micelle is small enough to get into this simple columnar cells, small intestine simple columnar cells. Okay, so it is very important to first form the micelles, and then micelles are small enough and to get into this simple columnar cells. And then let me go, let me clear this. Okay, let's just write down, let me redraw this. All right, microvilli, columnar cells. This is small intestine, so the chyme is moving this way. Okay, and then this micelle, oops, can be transported into these columnar cells. Then they have to somehow get out from this end, basolateral end, and then you're going to have the let me draw this first and then I'll talk about this. what it look like. This is the lacteal. Is the lymphatic capillary. Okay, so the idea is, if you have the fatty acid and small fat somehow can get through this basolateral membrane of the small intestine wall, and then it can get to the lacteal, which is the uh, lymphatic capillary, and then can be transported toward and then eventually get to the blood. Okay, that is how the fat can be absorbed. So we talk about the first step is to making the micelles. Okay, so once you have the micelles can get into this simple columnar cells of the small intestine. Once in the small intestine, the fatty acid, okay, so the micelle once inside this columnar cells, then this bile can be break down here and to release all the stuff inside of the center. Remember, we have the fatty acid. We also have the mono glyceride, cholesterol, all right, and what else? And bile. And now all this stuff, the fatty acid in these columnar cells can start to make monoglyceride again, okay? So as long as you have glycerol and fatty acid and enzyme, it can make a monoglyceride, okay? So now if you think about this molecule right here, okay, all of these molecules, different particles, and it's going to enclosed in the center and coated by proteins, okay, specific phospholipid proteins on the surface, okay, to make another circular structure. But this circular structure, it's much, much bigger than micelles because you're including more molecules and also bigger molecules. Just think about the proteins here. How big is the protein coat outside? Micelles, there's no proteins, right? So now this whole structure has another name. This one is called chylomicron. Chylomicron. Okay, so these chylomicrons can actually be transported through this membranous structure by exocytosis. This one has to be exocytosis. Has to be the process of exocytosis because this chylomicron is very, very big comparing to micelles. Okay, once the chylomicron is out, then it can get to the lacteals because the lacteals have this valves, if you remember, and which will allow bigger molecules to get in. So once the chylomicron get to the lacteal, and then it can be transported back into the lymphatic collecting vessels and the ducts and the trunk and ducts, and then back to the subclavian vein. Okay, then get into the blood. So in this way, we say the fatty acid and the small fat can be absorbed into blood. All right, so are we good with the fat digestion and absorption? Okay, we're actually talked about the physical digestion, which need help from bile, and the chemical digestion requires lipase, which is made in the pancreas. And the fat absorption, we have to first make micelles, you know, to get micelles into the small intestine, columnar cells. And then in the simple columnar cells, uh, the micelle will break down and restart making the small fat molecules, and also have the proteins, phosphoproteins, and to enclose everything in the center to form a chylomicron. Once chylomicrons are formed, it can be transported out from basolateral membrane by exocytosis, and then diffuse in, then transported back into the lacteals, and then back to the blood. So that is the uh, small fat absorption process in the small intestine. Okay, so I think I covered most of the digestion and absorption. So maybe let's do a little review here. Okay, so we started from the mouth. Let me go back to the slides. Okay. Um, okay. So we started from the right here. Okay. We start from the mouth, right? In the mouth, we say only carbs can be partially digested chemically, all right? Physical digestion takes place. No nutrients can be absorbed, right? And esophagus, no digestion, no absorption. And in the stomach, only proteins can be partially digested by enzyme pepsin, okay? And in terms of nutrient absorption, none of them, but alcohol can be absorbed in stomach. In the small intestine, this is the major location. This is the major location for chemical digestions, for carbs, proteins, nucleic acids, and fat. For all of these bigger molecules, biomolecules, chemical digestion, this is the most important location. The reason for that is the enzymes, rich solutions in the small intestine. The enzymes can be from pancreas or from the small intestine itself. Okay, so if the enzymes are from the small intestine, they are located on the microvilli. Okay, enzymes are located on the microvilli. So there are a lot of microvilli, increase the surface area, so you can carry a lot of enzymes. Because these enzymes are on the microvilli, we gave them a name called brush border enzymes, kind of like on the brushes. Okay, so the enzymes we talk about the proteases, we talked about enzymes break down the carbs, we talked about the bile, which can physically tear apart the fat particles. We talked about the lipase, which can chemically digest the fat molecules. And then in terms of absorptions, yes, all the nutrients can be absorbed in the small intestine. Okay, small intestine, like amino acids, glucose, those are easy to understand. We talked about facilitated diffusion before. Okay, that's how the amino acids and glucose molecules being absorbed. In terms of small fat and fatty acid, they have to be in the micelle form to get into the columnar cells, and then form chylomicrons to be transported out of the basolateral membrane of the columnar cells, and then get into the lacteals, and then back to the blood. So that is the process for small fat absorption. Okay, and then in the large intestine, no chemical digestions anymore, because the enzymes, the enzymes are in the small intestine. Okay, but in the large intestine, we do have a continuous absorption of water and bicarbonate ions. Okay, this is why you should have a good habit going for number 2, let's say every day. You should kind of like try to make a good habit, go to the bathroom about the same time every day, so you get used to it. But if you don't, if you don't go for number 2 for like say 3 days, and then it's going to get harder and harder to get rid of the waste, because the waste will stay in the large intestine, and the more and more water will be absorbed, and then it's going to be harder and harder to get rid of this dry waste. Okay, and also it's not good for your liver, because a lot of toxins, they already get to the large intestine, waiting to get rid of, but now you have these waste stay too long in the large intestine. Some of these toxins can be absorbed back, and then you just increase the workload for the liver again. Okay, so this is the digestion and the absorption process. And I like to kind of go over the enzymes and hormones we talked about today. Okay, so my board is not very big. Let me see if I can start writing all of them out. The first one we talk about is amylase. Amylase from saliva. Okay, do we remember that? Okay, and then let's change the color. Next one is a pepsin. Is from where? Stomach. And then next one is procarboxypeptidase. Number 4, chymotrypsinogen. Okay, and 5, trypsinogen. Are these from pancreas? Yes. Okay. All right. Do we have other enzymes from pancreas? Amylase, lipase, nucleases. Those are all from pancreas, right? Okay. Now, did I miss anything from pancreas? I hope not. Okay, let's change another color. Now we're talking about the brush border enzymes. Brush border enzymes, that's the enzymes from small intestine. So remember the sucrase, maltase, and lactase. Okay, 12 on this side. Proteases, do we know proteases? Amino peptidase, carboxy peptidase. What else? Dipeptidase. There's another protease in the small intestine. It's called enterokinase. I gave you this one earlier. What is the function? To activate trypsinogen to trypsin. Remember that? Okay. All right. So that's the enzymes from brush border part. Okay. Any other small intestine enzymes? Did I forget anything? No. Okay. All right, change the color again. Okay. So these are the major enzymes I want you to know. On top of that, I want you to know some hormones. One is gastrin, which is made by G cells in the stomach. If you remember that, if you don't, go back and review. Gastrin is involved in regulating a stomach function. Okay. Number two is called secretin. Okay. Secretin is from small intestine. Okay. Secretin can stimulate the pancreas to release more bicarbonate ion rich pancreatic juice. Okay. Number three is called CCK. This is made by small intestine as well. This one can increase bile release to enhance fat digestion. All right. So this is the list for enzymes and hormones I want you to know in terms of chemical digestion, chemically. Is there? I did not put a bio, did I? Okay, yeah. So these are for chemical digestion. All right. Okay. Enzymes and also the hormones which regulate the digestion process. Right. And for absorption, you need to focus on what materials can be absorbed in each part of a GI tract. Okay. And also for fat, absorption is a little bit more complicated. So you need to know the process, how to make the micelles, how to make a chylomicron, and how can a chylomicron being transported into lacteals and to blood. All right, that's it for this part of lecture. And this is going to be our last lecture for this unit and also for this semester. Okay. I will send you an email and before your final exam. Okay, so we can talk about the exam later. Alrighty, that's it.